let us start with a kind of introductory session on sequence alignment. So, people here are, who are in the call, and those who aspire to do studies, and like those who like to know the functions and the molecular function of any protein. I suppose understanding about sequence is more important. So one must have the knowledge to interpret any sequence, to identify how similar a sequence can be with other sequences, irrespective of factors. So you might not know under what criteria a sequence can be similar, or a sequence can be very much alike to some other random sequence that might bring new insights to you. So randomly a sequence from a human, fine. So as a sequence from a human insulin, a protein sequence, or a nucleotide sequence from a human insulin is kind of very similar to one from a dog. So sequence, understanding sequence, analyzing sequence are very big domains where you will be getting into newer insights of, to be precise, of organisms. So you will be like, so how we all know that we use lab rats, right? So for our experiments, for clinical, most of the clinical experiments in molecular biology or genome biology, we use lab rats. Why do we use that? So they are highly similar. Their genetic material, their genetic sequence is highly similar to that of a human, and that especially the coding regions, and that makes sense to test it on a rat. So understanding sequence is more important, fine, but this session is going to be a very basic introductory session, as constraining to a lot of limitations and as per the course structure. But if you find interest, so every single simple concept that we are looking, that we will be looking, or we are looking for these 3-4 days, you can specialize in any one of it. It is not necessary that you should know everything. You should know all these hands-on, or all these tools, and all the web servers which you have been exposed to. No, not at all. It is, again, as I told yesterday, it is up to your interest. What is your field of interest? You can, what is your domain of specialization? What do you want? Whether it is helpful for your research or not. So, so let's start. And today we will be looking at EMBOSS needle and blast. So again, we will be looking on a base, trying some hands-on at very basic level to put a simple understanding to you all. So we won't go deep, but hope I would just like to start with some background of sequence alignment before moving into hands-on with EMBOSS needle and blast. So yeah, so there are two types of alignments: global alignment and local alignment. So guys, global alignment covers the complete length of the sequence. So if we are comparing two sequences, we want to align two sequences of our choice, let it be a protein sequence or let it be a nucleotide sequence, a DNA sequence, we want to align it. When we go with global alignment, if the length of the sequence is 260 base pairs, or around 200 amino acids length, it will show an alignment of the complete 200 amino acid length. Or if both are of different sizes, it will try to bring out the picture of the complete sequence, how many ever number of sequences have been involved. So that is all about global alignment. And a Needleman-Wunsch algorithm is a very basic and technically strong algorithm for global alignment between two sequences. So here this deals with pairwise alignment, and whereas a local alignment, it covers significant parts. So it is not that you need to encroach the complete sequence. You have two big sequences. it will highlight the similar, the highly significant matching regions, with the like highly similarly aligned regions within the two sequences. So basically, global alignment is a tool to identify homologous sequences. That is, it is giving a complete coverage. So it will see whether it is highly similar or not. If it is highly similar, we call it homologs. Then on the other hand, local alignment tool generally tends to identify, its purpose is to identify the conserved and functional sequence motifs. So that is what it concentrates on, the parts of the sequences which are highly aligned. So I hope the image is describing things what I have told. Moving on to, so we'll start with local alignment. So there is some history about it. So it was introduced in 1981 and by Smith and Waterman. So that is, it goes by the name Smith-Waterman algorithm. So local alignment of two sequences is the alignment between the parts of the two sequences. So how does this happen? How does this is carried on? So before that, any alignment, we will be looking into matches, mismatches, gaps in the sense the insertion part or the deletion part, so which is not in the region of similarity. Considering all this, and but we are keeping in mind that we are not going to completely take the complete sequence. We will be concentrating only on the highly similar, highly aligned regions, and that is what local alignment does. So what does Smith-Waterman algorithm do? So this is, yeah, it's a complete technical part. It's a mathematical concept, like how to calculate. Like you might have tasks. So some people may say, okay, why, when we have software, why do we need to exactly know the algorithm? The idea behind teaching you, or the idea behind is exposing you to the algorithm is, in later stage there are people who in today's trend who are fond of learning machine learning, big data analytics. So I am one from the IT sector also. So what they do, they, when they continue with their research work, they tend to not to use these simple tools or the already available tools, but they try to create tools, or they try to create their own alignment tool of considering their particular requirement. So understanding the algorithm will always help you at any cost. So you, if you are a person who wants to explore through lot of lot of tools and the background of tools, so like basically the computational genomics part, so it is all about computational genomics. So at that point one must know the algorithm of whatever whatever technology or whatever idea or whatever to be precise, yeah. So this is one algorithm, so one technology that a tool uses. So one should be ready to explore all that. So that is why we have the technical content here. And yeah, so I would be not try not to explain all this in depth to not to confuse you. So I will go with an example. Yeah, so we see two sequences. So here we are going to try to put a local alignment matrix for two sequences. So we have the two sequences on the top and downways. So you see, before starting, we always have one row and one column for zeros. That is the initial start score. So generally in local alignment, it is always set to 0. In see, this is going to be the key differentiating part between the global alignment and the local alignment. In local alignment, the initial score, the initial calculation of the matrix always starts from 0. So that is why the complete first row and the first column is always 0, and then starts your matching column. So you are, if your sequence, you have two sequences, one is of length 5 and another is of a length 6. So your matrix will be of m plus 1 into m across n plus 1. So that is how it works. So how does the calculation happens? So you always need a matching score, you always need a mismatch score, and more importantly you need a gap score. So we'll just look how it is calculated. Yeah. So the very first item, it is A and G, right? So is everybody able to see the annotation I'm trying to make? Yeah, fine, thank you. So yeah, so it is A and G. So if A and G a match? No, it is a mismatch. And what is the score for mismatch here? We have a mismatch score of -1, right? So what we do is, whenever it is a mismatch, we used to add the mismatch score to the diagonal elements. And here. So what will it remain? Yeah, sure. I think I can, but I'm not sure if I could annotate in full screen. Let me check. Wait, I think I need to come out of annotation. Yeah, I have gone full screen. Okay, so is everybody able to see now? Yeah. So let me first clear. Okay, wait a minute. Clear all drawings. I have cleared all drawings. Yeah. So again, to start with, we have a mismatch. So the mismatch score, whenever you find a mismatch, it is always added to the diagonal element, right? So but it now it is the mismatch score is -1. Adding 0 and -1 will always give you, again it will remain -1. So whenever you get a negative score, it will be remaining 0. So that is what is mentioned here. It remains 0. So whenever the score is negative, it remains 0. But which -1 is contributing to this 0? Whether it is from here or from here. So to know that, whenever we calculate a score, if for a match or a mismatch, we will see which is the highest of this and this. Okay, I'll change the color. So we always will take the maximum score from this and this. Per se, now the updated score from the diagonals, because it is a mismatch, the updated score from the diagonal is -1 from here and -1 from here. So considering -1, 0 and -1, which is the greatest number? It is 0. Maximum number is 0, and that is how it goes. So the matrix is filled in the same way, in the exact way. If it is a match, okay, so I'll tell you what happens when it is a match. So let me take this table. So if it is a match, so C and C are a match. What always keep in mind that whenever you get a match, you have to consider the match score, which is +2, and add it to the diagonal element. Okay, so I use the wrong word. So you should take the the upper and your immediate side scores whenever it's a mismatch, or whenever it's a mismatch. Otherwise, you need to, whenever it's a match, you go with your diagonal element. So adding a 2 on 0 will give you 2. But what you have to do is, your side elements are not 0. You have a gap score, right? You have a gap score, right? So you have a gap score of -2. So always, whenever you find a match, add your gap scores to the side elements. That is +2 plus like adding -2 here. Wait a minute, let me clear. Yeah. So here it goes. We will have a +2 here and -2 2 here, and right. So again, after doing this, you will have to get the maximum score, which is the maximum out of these three, and that will be taken as your current score. And in the same way, the matrix has been filled, and the arrow mark, which is mentioned in this like in this table, right? So the arrow marks represents from which point that particular score has come. So 3 has come from its diagonal element because it's a match and it is highest score. So adding 2 with 1 will give you 3, but adding -2 with 4 will give you 2. So obviously 3 is the greatest number, the maximum score, and it is taken here. So the arrow mark represents the point from where that particular score has arrived. So this is the way we backtrack. Actually, you need not backtrack completely from the corner. You can backtrack from the highest score in the table. So you can see the highest score in the table is 6, right? So you start backtracking from 6 to the first match of your score. So the first score, actual score, comes here, right? CC, not this one, but this one. So 6, it is a match. Yeah, you have A and A, so it's a match. Then you get it has come from its diagonal. So looking at that, that is again a match. So that has come from its diagonal. Looking at that, that is a mismatch, and you can see that it has come from its side element, that is its side score. So we are doing that shift here. So since it's not a diagonal, that is, if it is not a match, definitely it is going to be either of the side. It is going to be this side of the score, or it should have arrived from this side of the score, and that will give you a gap. So we have a gap here, and then we get our match with TNT, and then we get our match with CNC. So this is the local alignment. That is, this is the region which is highly aligned, which might have a gap, but this is highly aligned within the sequences which we have aligned pairwise. Okay, so let me clear my annotations. Yeah, and moving on, so that was all about the local alignments with Waterman algorithm. This is for use for the global alignment. The highlight of Needleman-Wunsch algorithm is, even for relatively short sequences, there are lots of possible alignments. So what does that mean is, see, it is, can you please tell about gap penalty and gap extension? We have to manually do the scoring, or software or database will automatically do? Yeah. So software definitely will do all this. You have a lot of scoring matrix. You have a lot of choices for scoring matrix now. So everything is automated. You have the tools will do it automatically. So can you please tell about gap penalty? Yeah. So gap penalty is all about a score which you set. Generally, we always in to understand. Generally, we set gap penalty, how optimal local alignment we get, but we cannot set a very high, huge gap penalty, because when you set a huge gap penalty, it will tend to give you misinterpreted results. Your alignment might go wrong sometimes. Okay, for example, I'll tell you this case. Okay, so let me annotate to highlight that point. Yeah, so I'll tell you this case. Here we have a match, right? A and A are matching. So we get 3, and we got this 3 from its diagonal element. So generally, you have to get your match from your diagonal element. It's not a rule. Yeah, generally it is said so, because match scores are positives. It will give you increment in your score. Whereas the gap penalties, whenever you encounter a match, your gap penalty score will be reduced in the side elements. So as to understand, like this is just to make the match give an advantage to the match score. But what if I set a very low gap score? So if I would have set a gap score of -1, right, and a match score of +1, my score would be 2 here, and the gap score out of the gap I would have got 3 here, for example. Or because also see, changing the scores, obviously the other scores also will change. I'm just trying to bring a picture. So let's say the 4 is here already. Even if we change the scores, the 4 remains here. So out of which we will get a match score. If this is a match, we will add the match. We should get a score from here because the match contributes to increments and gaps contributes to decrements. But if we set match score and gap score in such a way that gap is so high or gap is so low, and we are misconfused, and even after a match the penalties are contributing to the score, then when we look at the alignment, we will get a gap even if it is a match. You understand? It can happen. It is not that it should not happen. It can happen even if it is a match. What is the gap here? What is this gap here? So okay, you see, right? C is a gap. So C is having a gap by the actual sequence CTCAA, and here you have CTAA. So the A should have been synced to C, and the last A should have been synced to, that is, this should have been here and this should have been here, right? So what happens here? So I'll tell you. Wait. C T C A A, and then you have C T A A. So the idea behind is, now this is also a possible alignment, right? So nothing wrong about it. This is also a possible alignment, but you calculate the matches. There's one match here, there is another match here, and then you have 3 matches and 1 mismatch. So calculating the score, now you can calculate the score. You have 3 match, 3 into 2, which gives you 3 into 2 will give you 6, and then you have 1 mismatch. So that will contribute to -1, and your optimal score remains 5. But look at the matrix where your optimal score, that is the highest score in the matrix, is 6, right? So this alignment is a good alignment, but not the optimal alignment. That is what the idea of gap here is. So what I do, okay, wait a minute. So I introduce a gap so as to improvise my match. So instead of 1 match without a gap, I'm getting 2 matches with a gap. So now my score, you can say, so now what will remain my score? I have 4 matches, I have 4 matches which will contribute to 8, a score of 8, and I have 1 gap. So that will contribute to -2, and my optimal score will be 6, which is the highest in the matrix amongst the 2 sequences. So I hope you understood the importance of gap, why we are using gap. Gap is nothing but it is insertion or deletion of a place. So sometimes I can insert an element so as to improve the match score. Sometimes I can make a deletion. So I have made a deletion here. Okay, to be precise, I have not made a deletion. I have made an insertion here. I have inserted a gap here to increase my optimal score, to increase my alignment and to find the optimal alignment. Sometimes you can also delete. So there can be cases where you will have to delete one of the amino acids. So that is, you have to consider it not be there, and then you have to proceed with your alignment. This is the idea behind gap. So moving on, yeah. So needle, but sir, will it not change the actual sequence alignment? No, it will not change. So that is what is the highlight of local alignment. Local alignment, you are going to see only the conserved region, right? Yeah, I'll tell you. See, in deleting, in the sense, it is not going to remove. It is not going to consider the exact pair. So if you have, okay, I'll tell you. 1 second. Yeah. So if you have C, T, C and A, and if you have some C, C, T, C, C, okay. So what it will do, it will try to bring a gap. That is, you consider it as a deletion. So deletion and insertion are just, it is a notion. If it is done in the second sequence, in the former sequence, you can call it a deletion. If it is done in the lateral sequence, you can call it an insertion. Basically, you are inserting or removing a gap. It is all about that. It is not exactly that you are removing the amino acid. You are just dispositioning the amino acid by introducing a gap or by reducing a gap. So I hope this will work, right? Or to be precise, I need not have this. I can push it further. So my alignment, so this is what I'm telling. I have dispositioned my alignment. That is it. I'm not changing my sequence. Yeah. So Needleman-Wunsch algorithm is, yeah, okay, coming back to that. So even for short sequences, right? Even for short sequences, you can have a lot of possibilities. So see, we saw a snippet of, considering 1 second, considering this as a global alignment sequence. So instead of a gap, let me introduce some other. So let me introduce this as A. Okay, and what I'm trying to do is I'm trying to look at the complete coverage, how exactly each and every amino acid is aligned, whether it's whether like majority of the amino acids or the majority of the base pairs or amino acids in the sequence are aligned as a match or not aligned. So that will show me how homologous, how similar that complete sequence is. So that is what is done here. Moving on to the algorithm of Needleman-Wunsch, it remains the same as it is, like Smith and Waterman, but the only thing is the initial scores are updated using your gap penalty score. So always my starting score at this point, let me annotate to highlight. Yeah, always my starting score at this point will remain 0. And from 0, we saw in Smith Waterman, it is completely zeros. It never takes negative numbers, negative scores. But Needleman-Wunsch algorithm, you are updating your gap score. So it is -2, it will be -4 next, -6 next, -8 next, and -10 next. And so will be your column. So the initial scores are updated using your gap penalties. So and the rest ideology is completely same. It is again your mismatch. You will have to add your mismatch scores, not the gap scores. You will have to add your mismatch scores to the side elements, and whenever you get a match, you will have to add your match score to the diagonal, and your gap penalty to the side elements. It remains the same, and again the algorithm is you have out of the three, which contributes the maximum score will be considered. So that was a quick view of what a local alignment and a global alignment could do. I would suggest you to like if you are students from bioinformatics domain, like third year or second year or final year students, I would suggest you to work on small snippets of codes. Try, if you are good enough, try to build small programs in R, Python, or maybe Perl, or whatever you use, to have a strong knowledge in working with algorithms. That will always help you. That will always like strengthen your technical part. So fine, now we'll go move into using EMBOSS. So EMBOSS is a pairwise alignment tool, right? So it has the background algorithm of Needleman, Wunsch, and Smith and Waterman. So we will try. I have already taken the sequence from, again, NCBI, which we saw yesterday. So I have just retrieved a sequence FASTA format from Parkinson's disease in humans, and another corresponding sequence, which is the same Parkinson's disease but with another organism. And I have taken musculus, that is a mouse. So let's proceed. We'll just see how it works. I think. Let me move to my, okay. So yeah, so I'll try to copy paste the link to be precise. I have copied and I'll paste. So fine, this is directly going to the emboss needle. Needle in the sense Needleman Wunsch, nothing else. So okay, I have come back to the pairwise alignment tool. So this is all about emboss. So it is basically powered by EMBL EBI, which is just like NCBI. It is a huge data repository database, and it has a lot of bioinformatics and services and tools within it. So one such thing is your pairwise sequence alignment, where your global alignment, you have two algorithms. So Stretcher is yet another algorithm which is modified. It's a modified version of Needleman-Wunsch. We will be looking at the Needleman-Wunsch version alone. And you have the local alignment, where you have again three types. Again, everything is the modified version and a changed version. So that is what I was telling to you, like you need to work if you are a person who works on with algorithms. So you can identify a new arrangement or a new parameter to highlight something, to provide some new insight. So that is why I was trying to stress on the algorithm at the starting part. So fine. So we will proceed with the Needleman Wunsch tool. So emboss needle. And I have okay, wait a minute. I will stop my sharing the screen. Just a minute. Yeah. All right. So let's start. I'm sharing my screen again. Yeah. So I have my alignment. Okay, I have already taken some samples to show you. So yeah, this is that particular sample. So this is the Parkinson disease protein from organism human I have taken. This is the FASTA format. So I'm copying this and I am going to have this as my sequence one. The you can see the angular bracket, right? So this first line of your FASTA format is always the name. It is the name annotation of your FASTA sequence. I can customize it to whatever name I want, like making it more informative. So sometimes I need not have the accession number, so I can just directly have it as Parkinson's disease protein. I need not worry about the number because it is just the person who has deposited, it is just for them. For us, it is merely a sample from Parkinson disease from human. Wait a minute, I think I can see. Okay, can we use this technique in plant disease identification in relation to data of disease? Yes, of course. You have lot of proteins, right? Proteins are present in every biological organism, so you can use. Provided you should be very particular of what particular region of your plant you are looking for and what kind of proteins you are looking for, and then you have to make a comparative. See, searching is yet another. It is a technique. It is not a simple thing. You need to be very clear of what you are searching, and only then you will retrieve your correct data, I would say, because every data is good. I won't say like you, but it is of not it is of no use to you if it is not a data that is going to satisfy your targets, right? So you be precise of what you want. You can definitely search for data for plant disease. You can make an alignment of different plant related disease or the proteins in plant and the same proteins in some other organism, and make a compare and contrast how that protein sequences are changed. Yeah, you can all do all that thing. So fine, I have pasted one sequence and I'm going with my another sequence from the other organism. Yeah, so you can see, right, enter a pair of protein. So if it is a nucleotide sequence, I would go with DNA. Or you, and also you can directly upload things as files. So sometimes these are very short, small, like short FASTA sequence, right? So you have sequences like for 30,000 base pairs, 35,000 base pairs. So you cannot copy completely everything and paste. So at that time you can have separate FASTA files and you can upload them directly. So output format. Now I think pairwise is all we have looked for, and all these are just formats and different kinds of alignment ways. So cluster W, I'll tell you, this is going to be easy because I like do not want to confuse you or go very deep into every single format. So one uh, like one very familiar format, just like pairwise, is cluster W, which is used for multiple sequence alignment. So here we are using two sequences, right? So for when we are using, so as this person, one second, let me, yeah, so as Arvind have asked, so can I use, can he use this technique in plant disease identification? Yeah, so you will have lots of protein sequences related to your work, your research. So you might have hundreds, you might have retrieved 100 sequences. So at that time, you will not be able to use pairwise. You cannot pairwise sequence every single. So how many iterations will you go? So that is a very hard task, right? At that time, you are going to use cluster W and softwares which will promote multiple sequence alignment. So that's just a small brief about it, and more options. Yeah, so this is what I was talking about. You have lot of scoring matrix. Okay, you have lot of different ways to score your matrix. As someone has asked, we have to manually do the scoring. No, the software does. So you have a lot of ways. And Blossom 62 is one very particular, one very common and most used matrix when it comes to pairwise alignment. So, or what does this matrix have? This matrix is not a very like what it is. It is not a secret or something. It is just mere combination of amino acids and some particular standard scores. So they have already given some standard threshold scores, and sometimes they calculate using these standard values. It is just standard values, and from matrix to matrix it changes. So what is the 62, what is the 60? These are just nothing but the mathematical annotations, and like there are some information, what kind of combination they have used. If they have used a 6 to 2 combination, that is how many reads they have considered, how many base pairs per alignment they have considered, all that. So this is just another information for you. Need not worry about this. So this is going to be a default thing, and now we are going to submit our work. Okay, so these are like very small sequences, right? So yeah, within seconds you will get your result. But when it is for larger sequences, you need not worry. You can always okay, I'll show you back. Let me not confuse you now. Yeah, so you can see, right, my Parkinson. Okay, so I should have named it better. Parkinson human and Parkinson mouse. So fine, no issues. So you can see, right, the alignment which has happened. So I have matches, and see, I have a mismatch here. Now you may question me, what about the gap? Where is the gap? You told that you should have given a gap, right? So that is what, you need not have a gap always. Gaps tend to promote your optimal score. So it always tends to like bring out the efficient alignments. And moreover, we are looking into a global alignment, right? So global alignment will cover top to bottom, the complete sequence. So this is showing how similar both sequences are. So you can see the identity is 173 upon 189. So the 189 is the length of the sequence. I mean, like both the sequences. Good, well and good. We have similar length sequences. Fine. So 173 upon 89 is the identity. Identity in the sense, how many mismatches, or you can see this. Okay, let me annotate. Okay, before that, difference between these formats. Okay, I think I have answered that question, right? So the difference between those formats is just they are different standards of calculation. So Blossom 62 is a matrix which has some particular calculation for set of combination of amino acids. So you have 20 amino acids, right? So 20 cross 20, there will be a matrix, and you will have some standard scores, and they will be using that score. So what does this double dots straight line mean? Yeah, so the straight line means match. What is the difference between sequence similarity and sequence identity? Yeah, I'm coming to that. So the double dots and straight line. So straight line means match. The dots and double dots, they refer to the mismatch, and the double dot is referring to the immediate. Okay, so the double dot is sometimes you can tend to, if you so this is a global alignment. Before that, let me make this clear. This is a global alignment. So in global alignment, we are trying to see the complete sequence, right? So that is what is the idea behind global alignment. But the double dot here and the single dot here, so the single dot represents mismatch. It's a pure mismatch, but the double dot is representing that if we are going to concentrate on a particular region, we can introduce gaps, and the possible gap position or the pivot for any gap will be that particular double dot. So let me annotate. 1 second. Yeah, so if I'm going to take this particular region alone, which is actually a local alignment, but yeah, to answer your question, so this refers to my mismatch, and this is also another mismatch, but I can, if when I'm seeing it as a local alignment, I can introduce this to a gap. So technically that is what is meant. So you need not worry like how can you introduce a gap in these two sequences. It is the general idea behind I'm trying to explain. it may differ, it may vary from sequence to sequence. So that is all it is about. And yeah, so what is the, yeah, to answer that, sequence identity is what is the number of base pairs, or the what is the number of amino acids are matched. So you consider the mismatches. So let me, yeah, so 1 2 3 4 5 6 7 8 9 10 11 12 13 14 15 16. Does it satisfy? So you don't have, you have like minus the 16 mismatches, you get 173. So that is identity. Identity is nothing but the complete match, the number of match in the whole sequence, like throughout the sequence. But what similarity is? Okay, so similarity goes by the score. Yeah, so we cannot see the calculation behind, but keep in mind we saw the algorithm, right? So based on the score, there is some optimal score for the global alignment, and this optimal score would have come from not only from the matches, that is the 173 matches. This might have come from this, this, this. I'm just randomly ticking, but yeah, this might have come from one of the mismatch or one of the possible indent, that is the gap. So that score tends to your similarity. So how similar the sequences? Yeah, it is so similar. So you can segregate regions. So this particular region is highly homologous, right? It is 100% match. So that is the ratio. They talk about the percentage in that way. Okay. So this is the pairwise alignment which we had done using needle and wunsch. So let's try something. Does both single and dot both represent mismatch in the sequence? Yes, both represent the mismatch. Both are representing the mismatch. The double dots can tend to be a gap when we focus on a particular region. So let me go with now Smith-Waterman. So we are moving on to this, and yeah, I have taken insulin sequence for this. So let me go with this. So this is an insulin snippet. It is not the complete insulin protein. It's an insulin snippet from Homo sapiens. All right. And this is going to be my insulin sequence from Myotis davidi. So I chose Myotis davidi because, okay, one second, I did not copy it properly. So Myotis davidi is nothing but bat. Okay, it is bat, and I chose bat because I'm a DC fan, so I just wanted to bring out some fun of DC comics here. So yeah, that's simple. Just randomly chosen organisms with no particular notion. So I just searched for insulin, and I got a snippet of Homo sapien, and I got a relative snippet from some other organism. So I'm going to try to run an alignment. You can see, right? These both are of not the same length. So let's see how this works when it comes to Waterman, Smith-Waterman. And yeah, I can also do this with global alignment. It is not that I should not do with global alignment. Basically, in global alignment, I will know how homologous these two sequences are. And now in Smith-Waterman, I'm trying to see what are the highly similar regions. That's it. So yeah, I'll go with the same. Let me have the same matrix. Okay. So I'll go with the, yeah. So here what I told, if it is a very large sequence, so if your sequences are large, you can always specify your email and title of your, so what, under what title it should be subjected to, and you need not worry. It is just a cloud operated tool, and you will get your you will get an email notification once the job is done and you have the result. So let me submit it now. Yeah, so my job is running currently. Yeah. Okay, so you can see here, right? I'm getting a gap directly. This is the idea behind Smith and Waterman. So we are looking at the region. So I can highlight some regions, right? So I will say this region is a local alignment. I can say this region is yet another local alignment, or some who can also consider the complete segment as a local alignment. No, 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 it is not like that. Yeah, you can use you can use any of the matrix. Only your score is going to change and nothing else. Your score will change. Probably I'll tell you, when you are like working on with bulk sequences, like very huge sequences, you will be choosing your matrix based on the combination. So I'll tell, okay, let me go back and let me tell you this clearly. So since you are interested and asked, I'll tell you that. Yeah, so these two remain my sequence. Now what I can do is I will go with Blossom 90, so which has some other set of standard calculation, so which has some other set of scores which will be involved. Previously I answered a question, right? I think you guys will be remembering. So why do we need a gap penalty? Okay, why are we setting it? So yeah, so this is the importance of having choosing a matrix too. So whenever you choose a scoring matrix, you should be aware of how like if you are going to go for extensive alignment in the sense you want very precise alignment, I would suggest go with Blossom 62 or lesser, like lower scores, because the more the scores are lower, your optimal score will be so precise and it will try to involve each and every mismatch, each and every gap. But what happens when you are taking scoring matrix with like of higher numbers? So these numbers basically represent how new they are and how like these scoring pattern will be inside the matrix. So what I'm taking Blossom 90, and when I submit the same thing, you might not see much variation in this result, but yeah, I'll try to bring a compare and contrast. Yeah, so this is what happens. So I hope you had seen the previous score, the change of score. So you might not see much of a difference, but yeah, but for a smaller snippet from 483 you got into 472, and eventually your percentages might vary from zero point 0.1 to zero point 0.2 percentage. Again, this is a small snippet. This will vary if you're going to work on with bigger data, bigger sequences. So that is the key idea, nothing else. You can choose any scoring matrix. Sometimes I'll tell you in studies, those who are working with sequence alignment and sequential studies, what they do is they'll try to build alignments with multiple scoring matrices. So multiple scoring matrices, and what they will do is they'll make a comparative analysis of the result. If they find much of a difference, they will pause. They will try to do some altercations. They'll try to retrieve some other sequences, more sequences to work on with. So basically you can use matrices for comparison too. So yeah, so that is all about Smith Waterman. So you can see the conserved, I can say that, right? So this is a particular snippet of, this is a local alignment. This can be a local alignment, or this put together completely can be local alignment. So this is what Smith-Waterman algorithm does. So we are looking into local alignment, and hence we extensively bring in the gaps. Let me clear all my drawings. Yeah. So guys, that is a quick, a quickie on EMBOSS. So yeah, we saw emboss needle, yeah, and emboss waterman. So let's move on to blast, using blast. So I think it's already time. We'll take, since we started late, we'll take some 10 to 15 minutes and we'll go with a quickie with blast. But I would like to tell you some basic information about blast. So I hope most of you would be knowing what is blast. Blast is a basic local alignment search tool. So it is not just alignment, it is a search tool. What blast does is, it now you have a sequence, you have a random sequence. Say, let me tell you. So I have a random sequence of say A G T C H K Y E W A G T Y, some random sequence. Okay, so consider this is in a FASTA format. I have this in a FASTA format. I have retrieved it somewhere or in some of my study. I identified the sequence. Now what I will do is, I would like to know how, where this sequence belongs. So in a clinical study, I have identified this sequence. I am trying to know where the sequence is belonging to, what the sequence is trying to tell. So at that point of time, I need to make a search with the existing database, which has a lot of sequences. And how will I search? Will I go line by line? Will I go sequence by sequence and try to align with each and every sequence? There are lakhs and lakhs of sequences. So BLAST does your job. So BLAST basically does a local alignment. It tries to highlight the highest conserved regions. It tries to bring in the molecular sequence motifs, similar sequence motifs, which other sequences have, and it will give you as a result. So there are different types of BLAST. We have BLASTN, BLASTP, BLASTX, T-BLASTN, T-BLASTX. So what is BLASTN? BLASTN basically compares nucleotide sequence with nucleotide sequences in a database. BLASTP, protein sequence with protein sequences from a database. BLASTX, it compares nucleotide sequence against protein sequence. So there is some internal translation happening. And what is T-BLASTN? So compares the protein sequences against 6-frame translations of nucleotide sequences. So what is the 6-frame translation? So what happens? I have a protein sequence and I'm comparing it with the nucleotide database, where I will try to convert my nucleotide sequences into possible proteins. So how are nucleotides converted into proteins? Does anyone has any idea? So I hope you will be knowing. People will be knowing. What is the idea? It's a very simple concept. How are nucleotide sequence? How do nucleotide sequence code for a protein? Genetic code. Yeah, ma'am. Yeah, of course, it is a genetic code. It will be translated, it will be transcribed and translated into a protein. Yeah, so okay. I'm not asking about the process. Okay, I'm telling now. See, I have a sequence. So when I have some combination of ATGC, some. So how will I identify a protein from this sequence? Yeah, exactly. Codons. Yeah, yeah. So that is what I was talking about. Codons. So T-BLAST10 works on the codon. So it takes a 6-frame reading. So basically, a DNA has 2 helix and 3 codons, 3 amino acid. 10 comprises codes to 1 protein. So 3 on both sides, both the helix. 3 plus 3 6. So that is what is the 6-frame translation, and it translates and gives you some kind of alignment results. And T-BLASTX is where you have nucleotide to protein. So what it does is, it initially translates all the nucleotide to proteins, then it goes to the protein database, then it again translates those proteins to nucleotide, and then it's a huge comparison. So you can see the query number. So one query. So DNA is one query, one search. Protein is one query, one search. When you go with BLASTX, that is, you have a nucleotide to protein. There comes the 6. You have 6 query searches. That is because of the 6-frame translation. In the same way, you invert it. You go with the protein to nucleotide. Yes, you get a 6-frame translation. But when you are using T-BLASTX, what happens is, your nucleotide will be considered as a protein, then to a protein to nucleotide. So that is what is, that's why it is giving a query search of 36. A lot of load to the software. Yeah, yeah, of course, it will give a lot of load, but you cannot take that load manually, right? So you need a software or a database to do that for you. So comparatively, it is a lesser kind of load. The load you are talking about. So yeah, so this is clear all my drawings. Yeah. So let me move on. So this is what is the idea behind BLAST. And yeah, you have a lot of special algorithms in BLAST. So Mega BLAST, RPS BLAST, BLAST 2 sequences, Genomic BLAST, Psi BLAST, Phi BLAST. So basically, it all started with BLAST. Okay, BLAST was an indigenous tool. It all started only with BLAST, and only these 6 types were inculcated, and these were randomly gone. And as you say, Pragya, yeah, a lot of load. Lot of load and lot of bulk data. It took almost around a day to identify like alignments for one particular snip of sequence, which I had given as an input. But now you have like fast processors, fast computers are so fast, and you have everything so fast, so things are done on cloud. So as I told previously, in EMBOSS, you give 2 bulk sequences for pairwise alignment. You need not worry if you are not getting in seconds, and if you want to go and have a quick snacks for 10 minutes, you go and come, it will be ready. So things are so fast, so you nowadays you won't call it a load, it is just a simple task. So, and then came improvements. So as there were improvements in the previous 2 algorithms, different facets of those algorithms, there are different forms of BLAST. So the Mega BLAST particularly does comparison for large data sets of nucleotide sequences. RPS BLAST, it does for combined conserved domain detection. So it will identify what domain does a particular big sequence belongs to. So which domain can be classified to. So BLAST 2 sequences. This algorithm is done for performing pairwise alignments for 2 chosen sequences. So you choose 2 sequences, you blast it, you can do pairwise alignment using BLAST also. Then genomic BLAST, yeah, for alignments against each selected organisms. So you will take human genome, you can take different genomes of different organisms which are available, and you are going to search your sequence against that particular genome. So that is a genomic BLAST. And Psi BLAST, which construct multiple alignment. Yeah, so Psi BLAST basically works with cluster W, as I told previously. So it constructs a multiple alignment, and from the matches, basically it does a simple alignment. Out of the matches, it takes the matches out, and it forms a multiple alignment out of the matches. So how many different sequences have the same sort of matches? And then Phi BLAST, it specifies a pattern that hits must match. Okay, so there are hits, there are BLAST will give you some hits. Those hits are highly aligned hits, highly aligned sequence, and Phi BLAST will specify a pattern. What pattern is contributing to this match? So it will highlight that pattern. So there are different facets of different special algorithm using the basic BLAST algorithm, right? So we will be looking into the basic one alone. So let me quickly start the BLAST. Okay. So what I will do is I'll directly search for BLAST. Yeah. Yeah. So this is the interface from NCBI. As we saw, EMBOSS was powered by it was under EMBL, right? So BLAST is under NCBI. So yeah, so you can find a quick introduction, and you can also learn more about BLAST here. So finds region of similarity between biological sequences, compares nucleotide protein sequences. Yeah, so that's what we were I talked about previously. So we'll do, so till now we saw protein, right? So let's try a nucleotide alignment. So we'll do a BLAST using nucleotide. I think I have a random sample. Yeah, so I have taken breast cancer gene. So it is another snippet. It is not a complete sequence of a gene. It's a snippet. It's an isolate from the breast cancer gene. So it's a partial gene, and let's go. I have taken this sequence and let's see how many alignments we are getting, how many similar sequences we are getting, and before that, how are we going to start with a blast? So I'm going to copy paste this here, and also as previously told, I can change the name, customize the name to our interest. You can upload it directly. So the job title will automatically be retrieved from the Angular tag or the FASTA sequence. If I'm not giving the Angular tag and I'm only giving the FASTA sequence, then I can manually type a job title which will be referred to as the name of the FASTA sequence. So yeah, so since this is a nucleotide nucleotide blast, we are using a standard database of nucleotide collections. So you have a lot of databases here against which you are going to run. Taking nucleotide, you are going to use BLAST10 program, and when you are using BLAST10 program, you should keep in mind that BLAST10 is directly comparing a nucleotide to a nucleotide, so there is no internal conversions. So please do not try to use a database which is completely onto proteins. Okay, so that will not give you fruitful results. So this is a standard nucleotide collection. It's a very dense and standard database. So we have chosen that is actually chosen by default. If not, you can choose it from the drop down. Then exclusion part. So organism, I'm not going to particularly look for organism. So yeah, there are people who will be working with specific organism. They will go with that particular organism. I want to look if the sequence is present in any of the database you have for a particular organism. No, but we need not have that. So I am not going with organism, but I would like to exclude the models and uncultured or environmental sample sequences. So why am I excluding these two? So these are models and sequences which are poorly annotated. I would not say they are less annotated, they are poorly annotated. The problem is the annotation done might not be successful or might not be accurate. So that would also disturb the result, that would like bring outliers to the result. So to avoid that, I would like try to exclude these sample sequence, these like models and uncultured sample sequences. And you might ask, if we are going to exclude this, then why should it even be involved, right? Why should this option itself be involved? Should this not have been a default thing? So the point is, sometimes there are sequences which you have no idea about. So out of clinical trials, out of some NGS sequencing, so next generation sequence analysis, out of the clinical trials, and you get a sequence. You are coming up with a sequence. You have no idea what that sequence is. Sometimes you will try to build upon the sequence. That sequence does not have a structure at all, an existing structure at all. It does not have a template at all. So at that time, we are tend to look at all possible sorts of sequences. So even if it is uncultured, even if it is poorly annotated, we can have an idea whether our sequence fall similar to those kind of sequences, right? So we can always try to say whether it is a hit, whether the sequence which we identified is working on wasting spending more time on that sequence, whether it is worth or not. So yeah, so the simple things like that. And then I am going to, okay, so this program selection. So here we are doing Mega Blast. Why Mega Blast? So we saw, right, Mega Blast is a special blast algorithm which is comparing DNA sequences, nucleotide sequences. So I said I would go with Mega Blast because it is specialized for nucleotide sequences and we are looking for a nucleotide hit. So we are going with Mega Blast, or you can go with BLAST10. Not a worry at all, because since this is a small sequence, it's a small snippet, you can go with BLAST10 too. So choosing algorithm here. Then before moving on to BLAST, I would like to show you the algorithm parameters which are set by default. So I have a maximum target of 100. So that is, it will show me top 100 alignments. So what if I say I want 500? So it will show me 500 alignments. So short queries. Yeah. So this thing is, I'm not activating it. What this does? So basically this is automatically adjust the parameters for short input sequences. So now my input sequence is a short input, right? And sometimes the blast, what will it will do is it will cut my sequence. Okay, some local, because it is again a local alignment search. So it will take the highly similar matching region and it will perform deeper alignment search using that particular sequence alone. So should I activate that? Yes, I should activate that, because only then I will get accurate results. What is my expect threshold? Okay, so before moving on to okay, we'll wind up, no issues. We'll just take 10 more minutes to explain what it and what this is, and we'll just look fastly into the results. But I would just like to tell what an expect value is referring to in BLAST. Okay. So basically the expect value is it is a parameter where the threshold is referring whether the output, whether the alignment result which we got has come by chance or it has some biological significance. So the lower the expect value, the higher the chances that there are like it is the output is of biological significance. So the point is, expect threshold generally tries to control the background noise. Here the background noise is, again, the uncultured sequences. So we might get sequences which have no domain at all, which have no proper annotation and no proper information at all. So yeah, so to reduce background noises, all such noises, we are setting threshold. So this threshold is at the default 10, and it can be varied by experiment to experiment and study to study. So your study, how will you calculate expect threshold? So again, you will be you should be knowing your NGS techniques to first of all identify your sequence from a clinical study, and then moving on what kind of threshold, what kind of expect value you could keep. And also you could iterate. It is not necessary that if you are not able to know what your expect threshold should be, you can iterate with different expect thresholds. You can go with 10, you can go with 100. 100 is too much. You can go with 10, you can go with 15, you can go with 20, and it depends. And word size, this is something very important we should know about. What it does is, now I have this big sequence, right? So my word size will be 28. So it is given as 28. The first 28 base pairs are taken. Then the second iteration, the next 28, third iteration, next 28, and then we will find and then we will see. So it will build a library of word queries, word searches, and then you will see the score. It will be scored again. Yeah, so that score will be corresponding to your expect threshold. If it is surpassing the expect threshold, it is well and good. You proceed with the alignment, you consider it as a good alignment, and then you proceed it for your local alignment or your global alignment, or you like take that and try to retrieve information from the database where it belongs to. And then yeah, scoring parameters. So I have different scoring parameters: +1 for match, -2 for mismatch, and gap. So my gap cost, generally it is not a score, it's a cost. Why do you call it a cost? Because at any cost, gap brings out the cost of, it decides, a gap will decide how a region is efficiently aligned or not. So generally we go with linear. Linear in the sense it will take, will it take consecutive 28? Only first 28 and then next 28. Yeah, it will, no, it will not take random 28. It will take continuous 28 snippets. So from 1 to 28, then 2 to 29, then 3 to 30, it moves on like that. Yeah, so there we go with the linear gap generally, to as to not complicate things. You can also take different extension values, different existence values based on your other scores, other queries, and if you are aware of how you choose your scoring matrix, then it is fine. Unless otherwise, going with linear will do. It is sufficient to go with linear to get better results. Okay, now let me go with my blast. So I'll quickly wind up with this search. We'll continue with tomorrow session. Yeah, so you can see, right, the page is refreshing. What exactly is being done behind is the algorithm is trying to make iterations, and it will just keep on refreshing on each iteration, and it will make a calculated time of what time it has tried to come up with the result. So that is basically the time log of BLAST. Yeah. So here you get your result. So I have my job title. Yeah. So I could have manipulated it. I went on with the name of the FASTA format. The program I have used, the database I have looked for, the query ID. This has been given a query ID. The query length. So the length of my query was 271. And you have some filter. You can filter your results. Again, if you want to look for particular organisms alone, you can filter it here. I want this much percentage of identity. So I am giving a range of 60 to 80. So I will get a result of only which has an identity of 60 to 80. Then e-value. So I will give e-value such that I will give the lowest possible range of e-value. Then I'll get the most significant results. Or if I'm giving the uh, largest range, highest range of e-value possible, highest in the sense high values of e-value. Results, I'll get the sequence. What is e-value? The expect value. I told, right, you have an expected threshold. Similarly, e-value is the expect value. This measure is a parameter that whether the hit, like whether these sequences are occurred by chance or they have some biological significance with respect to your input sequence. And the query coverage. So whether 100% of my query is covered or only a particular snippet of my query is covered, that is 90% of my query alone is covered, it is alone aligned, and then according to that match I'm getting something. Everything, every single detail is given here. And yeah, so moving on, we'll just quickly have a glance. I would appreciate if you guys try to like retrieve some sequences of some known genes of yours, some genes which you have come across. So I have used breast cancer. It is a basic example. Wherever you go, your professors or someone will actually give you. So if you have any idea of some other gene, or you can search with different queries of words, you can use choice of words, biological words, give a particular organism gene name, get some small FASTA format of protein or nucleotide retrieved, and try to blast and see what you are getting. Try to dig deeper. I would suggest. I would appreciate that. So yeah, see, I have top hits from Homo sapiens itself. So what this does is, I took a snippet from NCBI, right? It has given me all the immediate relative sequences from the NCBI itself. So this is the accession ID, which will lead me to the NCBI portal. And it has, so I can have 100% identical match, right? You see my expect value so low that these are significant. I won't say these have come by chance because these are significant. So let me scroll down. Yeah, so here you see I have 98.9% identity. So 1.1% of my sequence is ignored. I would say it has not been involved in alignment. Only 98.9% has been involved in alignment. And there I get, it is not only breast, it is an ovarian cancer too. So you get the difference, right? Till now we of only breast cancer, that is BRCA1, only that particular gene. When you get the identity low, when your E value increases, you will see the variation. It is not going to be only that gene. It can be susceptible to relative genes. It can be susceptible to relative organisms, relative organs, or relative biological molecules, biological sequences, everything involved. So generally, I'll give you a quick note like, okay, I'll not try to cover all these because it will take a lot of time. So again, this is just the alignment of each. So we have got 500 hits, right? So we will see 500 alignments here. So how the local alignment has been done. and we'll see 500 alignments here. This is all about the alignment. So what I will do is I'll give you a quick idea for before like you will be moving on to homology modeling tomorrow. So before that, one idea behind is whenever we are doing modeling, homology modeling. So homology modeling is done to build a structure. So you have a sequence for which there is no structure available, and you try to build a structure. For that, you have to do a blast search first. You have to take the sequence, you have to do a blast search, and your blast search, if it is giving you results of identity 100% or above 70% identities, then of course it has a structure. You can go with the structures which is above with the identity of above 70%. But when it is lower to 70%, when it is lower to in fact I'll say 70% is still a high value. When it is lower to 60 or 50, or like standardly we have it 70-30 ratio. So if it is less than even 30%, if it is touching less than 50%, you can consider that it does not have a defined structure, and you can go on building, you can go on modeling that sequence. So blast will help you to identify if a sequence needs to be modeled or if you can go with a existing structure. So winding up to that, let's wind up today's session, and hopefully I would like you to explore, because all these tools which we have covered for the past 2 days, they are highly exploratory. It is not that you need to have prerequisite knowledge about the tool. No, not necessary. You can dig, you can just identify what exactly each term means. So now Shivani has asked what is e-value. You can know what is e-value. E-value is just a theoretical concept. Okay, so you can just try to correlate with different try to retrieve some different sequences of your own choice. So I chose Parkinson disease today. That was my choice, and it was actually from my study. So yeah, in similar way, if you are interested in different facets of cancer, you can move on with cancer and try to retrieve some random sequence. Try to understand what blast is giving you, or what EMBOSS is trying to provide you. If you are able to compare and contrast with one or two sequences, or if you have some known statistics already of some alignment, try to take that particular sequence, try to take that particular information and try to sequence here. Try to see if you are getting the same result or similar result. That will help you to understand better, and of course it is going to be very useful for you if you are going to do a functional analysis based on sequence. So that's all for today. Thank you for being so patient and being so good audience. I hope I had throw light upon some new information to many. If not, extremely sorry if you already know, but I would definitely say that you will be you will be having more insights for the upcoming 2 days, because you are going to look on hands-on. You are going to get exposure of like comparatively more specific tool. So modeling is a very specific thing. It is not just retrieving and making an alignment like what we have done. So for alignment, we have a lot of things. We have blast, we have different variations in blast, we have emboss, we have different variations in emboss. There are a lot of other tools. But modeling is not like that. You need to be precise, and I hope you will enjoy the upcoming sessions also. Thank you. Thanks a lot for very patient listening. Not exactly. It depends on what is your what do you need. So the modeling will be done below 70% similarity. Yes, generally we prefer to do model before 70% similarity, because there might be a structure hidden which you cannot find, which you might not find within the sequence itself. But if you do, if you try to model that structure, you might come up with a very similar structure. Then you can go on with the same structure itself. So generally we use, you can also have a cutoff of 50%. So generally 50% and below is poor. It is poor. You cannot consider that as a hit, or you cannot consider that as it is a very identical structure. No, not at all. Per identity means matches in complete sequence. Exactly. Yeah, exactly. We saw the difference between identity and similarity, right? So here the identity refers to that. It is not that it is 100% identical or 100% match. Yeah, there are variations. So the percentage refers to the variations. So if your sequence itself is not query coverage is similarity. Exactly, exactly. I hope someone has got my point. Thank you. So yeah, so that is what, if your sequence itself does not have higher identity, your structure won't have. That is the main idea. So penetrating from sequence to structure, you will have lot of variations. So if your sequence is 100% identical, your structure will be 80% identical. Whereas your sequence itself is a 60 or 50% identical, how will your structure be? So that is the question mark we are trying to answer. We will be answering it tomorrow.